Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about DNA fingerprinting. Okay, now what is DNA fingerprinting? How we can use the DNA fingerprinting? What are the applications, procedure, each and everything we are going to talk about in this video lecture. My name is Muhammad Kamil Khan and you are watching my channel Kami Microbiologist. So let's start the video. Now in this video we mostly going to talk about the main content of this topic, and the main content are, first of all, we are going to talk about that what is DNA fingerprinting. We have to know about this. After that, then we are going to talk about the discovery of the DNA fingerprinting, means who discovered the DNA fingerprinting technique. After that, we have to know the basic terminology in DNA fingerprinting. Then the procedure through which, means steps are involved in the DNA fingerprinting, results which we obtain from the DNA fingerprinting, and what are the application of the DNA fingerprinting. First of all, let's start with the introduction of the DNA fingerprinting. Remember guys, this DNA fingerprinting is also known as DNA test. It is actually a technique which we can identify the person identity. Now how? Using the human DNA. Okay, so once we use the human DNA, this human DNA contain some specific nucleotide sequences that contain base pair that are unique to that individual. It means that the same nucleotide sequences or base pair will be not found in another human. Through this way we can identify the identity of a person. This is called DNA fingerprinting. Okay, this DNA fingerprinting was first proposed by a scientist named Sir Alec Jeffrey in 1984. Let's a little bit talk about this figure. So as you are seeing, this figure is actually the bends of the fingers. These are the lines that is present in our fingers. Remember guys, these lines are mostly different from person to person, and we know that there are millions of people in the universe, and each millions of people have different bends in their fingers, means these lines are different from person to person, and through this way we can say that one people is different from another. Through this fingerprint we can say like that. So this is a little bit introduction to DNA fingerprinting. Now let's talk some basic terminology which are used in DNA fingerprinting. We have to know about this. First of all, let's talk about that what is DNA. As all of you better know that DNA is nothing but these are a molecule that is present in every cells, and this molecule is actually contain basic information. Now this basic information mostly obtained from the parents to the offspring. So we can say it is actually the hereditary material that can store all the information. So when we know the person identity, so we mostly know the person identity by this DNA. Okay, then remember guys, inside the DNA there is 99% portion of the DNA that is similar in all the person. Okay, so we can say in all the person the 99% area of the DNA is similar, but 0.1% are different. Okay, it means that the 0.1% area in the DNA are different of every person, and it contain repeated sequences. Through these repeated sequences are actually different from person to person. So this is a little bit another most important information to you guys. Then the second term is SNPS, Then we have single nucleotide polymorphism. What is this? These are actually the sequences in the DNA. Okay, remember guys, these are what? These are actually the sequences in the DNA that are actually different from another DNA through one nucleotide, okay, or one base, as you can see. So let's suppose this is the sequences of DNA of one person and this is the sequences of DNA of another person. Both are similar to each other, but a small variation can occur at a single nucleotide, as you can see. In this case we have C, but in another case we have, instead of C we have what? T. So this is called single nucleotide polymorphism. When single nucleotide is changed by another base pair, we call that single nucleotide base pair or single nucleotide polymorphisms. Okay, so we can say that at every person, although the DNA are simple but similar, but still there is one nucleotide changes occur. Okay, now the second terms that we have to remember in DNA fingerprinting, that is called RFLPs. What does it mean? It means restriction fragment length polymorphisms. Now what is restriction fragment length polymorphism? First of all, let's see the diagram, then I will tell you that what is this. Remember guys, this is actually the first DNA sample of one person and this is the DNA sample of another person. Okay, now these are the sequences, as you can see. Okay, what we have mostly, we have an enzyme which we call that restriction endonuclease enzyme. That enzyme have the ability to cleave the DNA at a specific point. We also call it molecular scissor. So they have molecular scissor like activity, so they can cleave the DNA at a specific point. So this restriction enzyme is actually first of all isolated from a bacteria which we call that E. coli. Okay, so what happened mostly, this restriction enzyme had a specific site to cleave the DNA at that point, at that point, and at that point. Due to this way, what we get the fragments of the DNA, as you can see. Now once we get the fragments of the DNA, the length of these fragments of the DNA are different from each other. As you can see, this is the first fragment of the DNA, second, third and fourth. So the two length of the DNA fragment are are same, but the other two are a little bit varied from each other. In the second case, although the sequences are same, but just a little bit is changed, and that is G. So in this case we have G, but in this case instead of G we have A. Due to this single nucleotide polymorphism, due to this single nucleotide changes, what happen? The same restriction enzyme will cleave the DNA, but it will not generate the 4 fragment as in the case of DNA 1. It will generate only 3 fragments, as you can see, and these 3 fragments of the DNA are also different from this DNA number 1. Okay, so it is due to this one nucleotide, as you can see. RFLPs means, so now let's talk about this. These are actually the DNA fragments that are generated during restriction enzyme activity, and these fragment DNA have different length. So remember guys, the length the fragments of the DNA, as you can see, having different length, it must be different from another person as well. It doesn't mean that the fragments of the DNA which we obtain after restriction enzyme, it will be similar to the second DNA of another person. Now it must be different from another person. This is called RFLPs, restriction fragment length polymorphisms. Okay, third we have satellite DNA. Now what is satellite DNA? All of you better know that inside the DNA, what we have two region. One region is the coding region, another region is the non-coding region. Coding region is specially responsible for the protein synthesis, while non-coding region is the junk portion. It is not responsible for the protein synthesis, we can say like that. Mostly these non-coding sequences of the DNA have a special type of base pair which are repeated many times. So remember guys, what we have the non-coding sequences in the DNA that have base pair which are repeated many times. This is called satellite DNA. Okay, so I hope you guys get it about that. Now then we have ministatillite DNA and microsatellite DNA that are the type of the satellite DNA. This ministatellite DNA is also called as VNTRs, variable number tendon repeats. What are this? These are actually again the type of satellite DNA in which we have non-coding sequences which have the base pair, but these base pair are 5 to 10, 5 base pair, or we have 10 base pair. But these 5 and 10 base pair when we have, so these are repeated so many times, or many times it is repeated. We can say 50 to 1500 times it is repeated. Okay, this is called ministatellite DNA. Let's understand it with an example. As you can see, these are the sequences of the DNA of one person. So as you can see, we have what? We have G, C, A, T, A. So these are the one, and then the same sequence is repeated here as well. As you can see, G, C, A, T, A. Again it is repeated, G, C, A, T, A. So remember guys, if these sequences, these are what? These are the base pair, 4 base pair. If these 4 base pairs or 5 base pair or 6 base pair, if it is repeated so many times, we call it ministatellite DNA. And we also call it variable number tendon repeats. These are the variables, variables base pair, and it is repeated many times. Okay, and it is mostly repeated not less than 50% and not more than 1500 times. Okay, so remember guys, these number repetition is different from person to person. If let's suppose in one person it is repeated, let's suppose it is repeated 100 times, so in another person it will be repeated 12, we can say 150 times, 160 times. So from person to person it is repeated, the repetition is different, we can say like that. Okay, so this is called ministatellite DNA or variable number tendon repeats. Second, we have microsatellite DNA, or we can say simple tendon repeats or short tendon repeats. We can also say simple tendon repeats. So in these are the same as we talked in the ministatellite, but in this case we have what? We have the base pair, base pair number are less as compared to variable number tendon repeats. In this case we have 2 base pair or 4 base pair that is repeated many times. Many times means not less than 5 times, not more than 500 times. So remember guys, if we have the DNA sequences, non-coding DNA sequences in which the base pair is repeated many times, like not more than 500 and not less than 5, we called it microsatellite DNA or simple tendon repeats. As you can see, this is the example: ACG, ACG, ACG. So 3 base pair, and it is repeated so many times. Here I just repeated 3 times, but it is repeated so many times. This is called microsatellite DNA. And again, this sequences is not similar in another person DNA. Let's suppose their DNA 1, so the DNA 2 will be different from this way. Although they have the same sequences, but the repetition will be different. If it is repeated 10 times, so the other will be repeated 20 times, the other will be repeated 6 times, the other will be repeated. So the repetition is actually different from person to person. Okay, that's how we can identify one person that is variable from another person, through this repetition sequences of the DNA. Now let's talk the procedure of DNA fingerprinting or DNA test. So to perform the DNA fingerprinting, we have to know all these steps. These are actually the most important steps that are used in the DNA fingerprinting. First of all, what we have to do? First of all, we have to extract and isolate the DNA from the sample which we collected from the crime scene or from anywhere. Okay, let's suppose we have to collect the sample, and from this sample we have to isolate and extract the DNA. The sample will be, doesn't matter, it could be a blood, it could be hair, or it could be any tissue. Okay, once we collect the sample, so from that sample we have to isolate and extract the DNA. After that, once the DNA is isolated, then this DNA have to be digested using restriction enzyme. Means it will be converted into fragment by restriction enzyme. Once the DNA are converted into fragment, we have to amplify this DNA. We have to make the copy of this fragment DNA using PCR technique. After that, we have to run this DNA by making a gel which we called it polyacrylamide gel, or SDS page, sodium dodecyl polyacrylamide gel, or agarose gel as well. Once the gel is prepared, so then we have to perform a technique on this gel which we called it gel electrophoresis, in which the sample that contains the DNA is separated on the gel, which we can see on the gel in the form of bands. After that, what we will do, we have to denature the DNA using a solution which we call it sodium hydroxide or a potassium hydroxide. Denature of DNA means it doesn't mean the degradation of DNA, or we can say the digestion of DNA. It means the two strands of the DNA should be separated using these two solutions. After that, we have to perform a technique which we call it southern blotting using nylon membrane or nitrocellulose membrane. And once we perform this technique, then another technique is hybridization. Now hybridization, in case of hybridization, we mostly use radioactive probes, which are the non-sequences having a radioactive substance that can give fluorescence when it binds to the complementary DNA sequence. Last, we have autoradiography, on which we use X-ray film, through which we can easily visualize the DNA in the form of bands. So this is actually the procedure of DNA fingerprinting. Now let's talk the explanation of these things. This is actually just the step-by-step process, but we have to explain it to understand the process easily. So let's explain this process. Now to explain this process, first of all, what we have to do, if we want to check the DNA fingerprinting of a person, if we want to identify the person identity, first of all, what we have to do, we have to take the blood sample, tissue sample, or hair follicle. Let's suppose once we take it, after that we have to isolate the DNA. Now here the question arises in our mind that how we will isolate the DNA from this blood sample or any other sample. How we can isolate, because the DNA is present in the nucleus and it's very difficult for us. We have to treat this cell using detergent solution. So once we add the detergent solution into this sample, it will rupture the DNA, and when the DNA is ruptured, so after that we get the DNA. Once the cell is ruptured, so after that we get the DNA. Okay, so once we get the DNA, after that, as we cannot see it, what we will do, we have to centrifuge this tube. Once we centrifuge it, so we get two layer. The one layer is called supernatant, and the other layer is called pellet. pellet is mostly contain the heavy particles, where the supernatant is contain the lighter particles. So we do not need the supernatant because supernatant contain the all the debris, but the pellet contain the DNA. So we will discard the supernatant and resuspend this pellet into another into buffer solution. Once we will resuspend it, so then we will leave it. After that, what we will do, we have to add a restriction endonuclease. By adding restriction endonuclease, it will cleave the DNA or digest the DNA into different fragments. As you can see, these are the fragments of the DNA which we cleave it using restriction enzyme. After that, we have to amplify this DNA using PCR techniques, as you can see. So these are the PCR techniques. Remember guys, in the PCR technique we mostly make the millions from one DNA we get millions of DNA copies, as you can see, from one fragment of the DNA. In this case, what we do, first of all, we have to denature the DNA, means separate the strain. After the separation of the strain, so then the primer will be added, and then this primer is elongated. Then again the DNA is melted, again the primer is added and is elongated, and the process is repeated so many times, and we get the copies of millions copies of the DNA. This is called PCR technique. We have to prepare agarose gel. Now how we will prepare it? It's a question arises in our mind. We have to take agarose gel, and it's mostly present in the form of powder. We will mix the powder into double distilled water. After that, by mixing it, so we will have to heat it by boiling. Once it is heat boiled, so then all the content will be easily mixed and dissolved. Now after that, this is a tray of the electrophoresis in a machine or a device. So in this device, what we have to load the gel, which is in a liquid form. After a few minutes or hour, after 1 hour, what happen, this gel will be in this liquid solution will be in the form of gel. Before that, we have to add a comb. This comb will create the wells. As you can see, these are the wells which is created by adding the comb. So then it will be gently removed once the solution are in a gel form. So once this process is done, what we have to do, we have to take the sample from the PCR machine, which is the DNA sample. We have to take it, and then we will load the DNA sample into one of the well. If we have one sample, so we have to add it in one wells. If we have 2 3, so how, so this then for example, we have 10 samples, so we have to make 10 wells in this gel. Okay, so this, once the DNA is loaded, after that what we will do, we will have to provide the electric current, or the process called electroporation. Through this electroporation, what happen, the DNA sample which we loaded here, it will be started moving, and it will be moving, and the DNA fragment which is heavy, it will be moving less, and the DNA which have length is small, so it will be moving fastly, or the DNA which having low weight, it will be moving fastly. Okay, and what we get the bends, as you can see, but we cannot see it. Okay, remember guys, in the gel you cannot see the bends. You cannot see the bends, means you cannot see the DNA in the form of the bends. What you will do to check the bends, after that you will have to use a technique which we called it southern blotting technique. In this case, you will use a filter paper which we called it nylon filter paper. You will have to apply it on the gel. So once you apply it on the gel, so it will receive or catch the DNA bends which we cannot see it. So the DNA bends will be received or transferred to this nylon filter paper. Once it is received into this paper, so then we have to put it into another solution which can contain the probes. Probes are actually the non-sequences which contain a radioactive substance, and these non-sequences, if let's suppose it is have to binds with the DNA. Before that, I forget one thing, that this gel contain sodium hydroxide that will denature the DNA, means separate the two strand of the DNA, and after that we have to shift it from the nylon to the nylon filter paper. After that we will load this nylon filter paper into another solution that can contain probes. The probes, which is the noun sequences, will bind to the complementary DNA, and once it will bind, so what happen, it will produce the color. So once the color is produced, so what we get the DNA. Okay, this is how we can get the DNA in the form of the bands. So still we just see the color, we not see the bands. So how we will check it, the bands? For that purpose we perform another technique which we called it autoradiography. So how to perform autoradiography? In this case we can take X-ray film and we will put this X-ray film onto the nylon filter paper, and once you will put it, so what you will get, you get the bands, as you can see. So these are actually the DNA. What? These are the fragments of the DNA. So you can easily visualize the DNA fragments in the form of the bands using X-ray film. Now this is your identity, as you can see. Now this is your identity, and this is not similar to another person. It will be different from person to person. Okay, so this is how we can perform the DNA fingerprinting technique, through which we can mostly identify one person from another person. Now let's talk about the applications of the DNA fingerprinting. Remember guys, we have different applications of the DNA fingerprinting. We mostly use it to solve the crime, like in case of murder and rape, because we better know, let's suppose if someone get murder or if someone get raped, so we can easily identify that who actually, which person is involved in murder or in the rape case. We can easily identify it using DNA fingerprinting technique. Using DNA fingerprinting technique, we can solve the problem of paternity. Okay, and means we mostly solve the paternity problem. Paternity problem means that, let's suppose there is an issue between the family, that there is a baby and everyone are fighting, that someone says this is my baby, someone says this is my baby. So to solve this problem, we have to perform DNA fingerprinting or DNA test. So through this test, we can easily identify that who is the parents of this baby. Okay, it can also identify the personal identification, and it is also used to identify a disease which we can say AIDS disease. Now how? There is a question arise in our mind, that how? So as you can see, these are the bands, and these are the X-ray film. Through this X-ray film we can visualize the DNA in the form of the bands, as you can see. This is the person number 1 DNA, bands 2, 3, 4. So 12 person having 12 bands, as you can see. Okay, and in this case, if you see 3rd and 10 person bands are similar to each other. We will talk to it later, but this is, let's suppose, how the DNA bands is. Let's talk that using DNA fingerprinting, how we can identify a suspected person that are involved in murder case or in the rape case. How you will identify it? For that purpose, what you will do first of all, you have to take the blood sample, as you can see. Let's suppose at the area where, there is an area where the dead body is present. We don't know that who actually killed this person, who are involved in this murder. Okay, we don't know about that, but we just get some blood sample. Okay, we get the blood sample from that area. So we have to collect the blood sample, or we have to collect the hair follicle or the tissue which is actually this tissue and this blood sample or this hair will be the murderer. Okay, these are the sample of the murderer. Once we take the sample of that person, what we will do it, we have to isolate the DNA from this, as you can see. Once we isolate, and we have to proceed it, as you can see, which we already talked. So once we proceed it, so what we will do, we get the DNA bands, as you can see. Now these bands are actually, once we take the blood and we proceed this blood sample from these all procedure which I already talked, so we get the bands of the DNA. Now these are the bands of the murderer, means the person who killed another person. These are the bands of that person, because that person, what we can say, their blood are dropped at that area, so we collect the blood from that area, and from this blood what we can get, we get the identity of the person in the form of the bands. Now how we can say that, let's suppose if we have doubt in 3%, as you can see, person, let's suppose these are the bands of the 3%, person A, person B and person C, and these are the blood sample which we collected. Okay, these are the blood sample which we collected from the crime scene, and after that we get the bands, and these are the 3% A, B, C. These 3% we are doubt on these 3%, that from these 3% one of the person is the murderer. So how we identify it? As you can see, this DNA bands are not similar to this A person, but it is similar to this B person, and again it is not similar to this C person. What we get? We get that this blood sample is actually the person B blood sample, and it means that person B is actually involving in the murderer case. Okay, this is how we can solve the murderer problem or the rape problem. Okay, and easily identify the murderer. Let's talk about that how we can identify or solve the paternity issue. For that purpose, you have to take the blood sample from the baby as well as the blood sample from their parents, because the DNA will be transferred from the offspring. Okay, we get the DNA from the offspring. So once you proceed the steps to all, these are the procedure. Okay, so once you proceed the blood sample through this process, so what you will get at the end, you will get the DNA bands. These are the DNA bands. Again, these are the DNA bands of the baby, as you can see. Now these baby DNA bands are not matched with the person A, but it is matching with the person B. It means that this baby, their parent is person B. So person B is the parents of this baby. This is how we can identify or matching the DNA with baby with their parents. This is how we can solve the issue. Another thing is the person identity. So it is very simple. The person identity, as you can see, these are the person. This is one bands, this is another bands. So one bands of the DNA is not similar to another DNA bands. It means that this is the identity of the one person which is similar to identity of another person which have different bands, as you can see. This is how we can say that we can easily identify the person identity. Now using this DNA fingerprinting, we can also detect the AIDS disease or different kinds of disease. Now how we have to know the RNA or we have to know the HIV virus sequences. Let's suppose this is what these are the HIV viruses DNA bands. So these HIV virus DNA bands are not simple to this disease, but it is simple to this disease. And this B is what? This B is actually AIDS, and these are the HIV viruses. So AIDS and HIV are similar to each other, so then we will say that this person have AIDS disease. 100% is confirmed that this person have AIDS disease. This is how we can detect the disease. So that's all about the DNA fingerprinting and their application in detail. I hope you guys get it about that. Thanks for watching.